share my screen here and show you, while we're waiting, feel free to go ahead and scan this QR code or type this URL in your browser. This URL also is being shared in the chat box. So you should see in GoToWebinar, you will, in the chat box, you will get a link soon with the link above here if you are not able to type it or scan it with a QR code. And this will take you to a poll. As you can see here, people are already starting to answer. And the question we're simply asking, we've been asking this in all of our webinars, is which database do you use? This is a multi-select, so you can select more than one database. And that just gives us an idea of what databases people are working on who are attending a particular webinar. So if you can go ahead and do this, that'll be much appreciated. The link is in the chat box and also posted up here. Also, while we're waiting, just some basic housekeeping for GoToWebinar. In your control panel, there is a questions section. This is a place where you can type questions. So throughout the webinar, feel free to type your questions there, and we will answer them as we get them. And if needed, we will ask the question orally to everybody, and the presenter will go ahead and answer it as well to everybody. So depending on a question, we'll take care of it that way. Okay, let me go back to the poll. Let's see what it's looking at. So again, feel free to answer this. I'll keep this running throughout the webinar so that you can answer at any time if you want. And if we have time in the end, we'll show it again. Great. So go back here. So today's webinar, in case you don't remember, or if you weren't sure, today's webinar will focus on MapVEu, which is a nice population biology mapping system and filtering system as well. Today's presenter is Sam Rund, who's based at the University of Notre Dame. He's part of the VEuPathDB project, and he'll be walking us through various aspects of this MapVEu tool, which is very exciting. Right now, it's available in vector-based, but will become available in all of our resources over time, and has some very nice functionalities. In addition, before we get started, I wanted to make sure that you are aware that you can access on our beta sites. So if you go to beta.veupathDB.org or beta.vectorbase.org or beta.plasmodb.org, depending on your favorite resource, under the help menu, you will see that there is learn how to use VEuPathDB. And this offers you a number of options, including various learning materials, access to previous workshop material, and these will be updated over time. But what I want to highlight here is our webinars. We've been running weekly webinars for a few months right now. You can explore these further, so you can sign up for new ones, upcoming ones. Today's webinar is the MapView one. And the nice thing about this is that we have all of our previous webinars are listed here, including links to the recordings. So for example, yesterday's webinar, which was a general introduction to the beta sites, is posted here, and you can go to YouTube and watch the webinar if you missed it and you want to go back and get more information. And of course, all of our previous webinars are listed here with links to the recordings. Okay, so without further ado, I think we have a good number of attendees right now. Sam, I'm going to go ahead and turn it over to you and make you the presenter.
Thank you, Omar.
Great, and we can hear you well. We don't see your slides yet. Okay, perfect. Now we see it. Thank you.
Thanks. So MapVEu, which was previously known as the vector-based pop biomap, is currently only available on vector-based, as Omar said, but we continue to move forward as a combined VEuPath BRC, and we'll bring MapVEu to other databases in the VEuPath family of sites. You can access the tool by clicking Tools, MapVEu, and this takes us to our MapVEu interface, which is, as it sounds, a map with data points across the globe, and we call each of these circles markers. In this first default view, we are showing all the geospatially linked data that we have, which, as you can see here in our right-hand drawer, ranges from everything from abundance data to the locations of whole genome sequencing projects. Now, each of these marker points acts as a pie chart and shows the distribution of available data types at that location. And we can zoom into a map by drawing a rectangle around an area of interest while holding down the Shift key, like this, or by double-clicking on a marker point. So I'll double-click here on Tanzania. And if you noticed, as I double-clicked on a marker point, the marker points disaggregate, and I can see higher and higher spatially resolved data. And I continue to double-click on the marker points to further disaggregate the marker points until I get to a marker point that has this thumbtack here in the corner of the marker circle. And this means data is now displayed at the highest spatial resolution that we have and cannot be further disaggregated. I mean, you can, of course, zoom in farther, but it won't disaggregate into smaller points anymore. Now, going back to the right-hand drawer, we can, for example, instead of faceting by data type, we could facet to change and see things by species. So I'm going to zoom out a bit first and click Optimize Colors to make them easier to see. And now the pie charts now the colors that make up the pie charts represent the species distribution of the data instead of the data types. Now, I do want to stress at this point that all the marker points represent the distribution of the available data we have in VectorBase, and not necessarily the actual real distribution of the ground of any particular experiment. So just keep that in mind. It's a count of how much data that we have. Now, most of the data we have in VectorBase PopBioMap is mosquitoes, but there are other species represented. And after my presentation today, if you have any good spatially resolved data sets that you think might be useful to have in MapView, please do get in touch with us. So I'll transition here now a bit. For 5 of our data types, which you can see here from this Views drop-down selector, we have built special filters and tools for interacting with this data. And I'll start with the abundance view. So I'll click here, and I'll click on full map or minimum zoom so I can see the entire world. And much of this abundance data that we have in VectorBase is collected from US mosquito abatement districts as part of their regular ongoing surveillance efforts. But we are making a big push right now at VectorBase to get African longitudinal population surveys too. And so as I show you this data, if you have or are aware of such data sets that we can have access to, the raw data, please do reach out. We would love to chat. So now I'll zoom into Florida, again, by clicking Shift and drawing a box. And Florida is where we have the highest concentration of mosquito surveillance in the United States. I'll click Optimize Colors. And Optimize Colors optimizes the variable colors based just on those variables, in this case species, that are displayed on the screen right now, as opposed to the entire world without any filters. Now, the numbers in abundance view that you see inside the markers, these represent the number of specimens collected at that location across all the time that we have at that location in VectorBase. And I can click on a marker point again, as we did before, and see the species breakdown at that point over here in our left-hand panel. So I'm going to zoom in here on the area around Naples, Florida. And once I do this, I'm going to start to filter data because I want to look for a specific subset of data. We like to think of PopBioMap as kind of a data discovery engine. So here in this filter box, I'm going to filter, I just want to see mosquitoes of the genus Anopheles. So I can start to type, and you'll notice we have the predictive text here. So I'll click genus Anopheles. And now you can see here in the right-hand drawer that we only have Anopheline species listed. And again, I'll optimize the colors to make it a little easier to see. Now, a word of caution on our predictive text, because this trips up a lot of users. If I start to type something, you can't just push enter when you see the thing you want. You do have to actually take your mouse and click on the term you want, or it will produce some unexpected behaviors. And now that we have two terms here, in our filter box, this is interpreted as a Boolean or, as long as the terms are the same. So if you have two species here, or species groups, it means that it will display all data that is genus Anopheles or Aedes vexens sensu lato. And I can remove that term that I just added by clicking on the X to go back to just seeing Anopheles data. Another way we can add search terms is using the faceting box in the right-hand drawer. So I'm going to switch the right-hand panel, right-hand drawer to collection protocol. And then if I just click on CDC light trap, that will add it to my search filter box. So now I'm looking for things that are, because collection protocols and taxonomic group are different, this is interpreted as a Boolean and. So I'm only looking at data that's genus Anopheles and caught by CDC light trap. So I will switch back to my species facet, optimize those colors again, and I am going to zoom in this time by just double clicking. I'm going to click on this point here. And instead of just looking at the pie chart, because this is the abundance view, we built a specialized search tool. And I'll click on our timeline view. And so I can see how many mosquitoes were caught over time at that specific point. And right now it's by default been to yearly, but I can see much higher temporal based data by clicking here on VEuPathDB. I can make that bigger. So I've expanded it. And so as you can see here, you can see the raw trap level counts from a long-term collection of mosquitoes. And as we explore the other tools here in the left-hand panel, by going to the table view, you can see all the data we have collected has rich metadata associated with each of the records. This includes what trap was used, what attractants were used in that trap, how many individuals were caught of a particular species, what sex was caught, what development stage, and even what method they used to species identify the mosquito or the vector, since sometimes it's more complicated, or species complexes, etc. This data can be downloaded using the download pane, and you can select which data you want to download. For example, data matching search, so that for anywhere in the world that's genus Anopheles or CDC light trap, in the example I have on the screen. Data visible on the screen. So this would just show the data from those 24,000 mosquitoes that were collected that we can see on the screen right now. Or click on one given point and just download the data from that particular point. You can see that these are all the metadata fields that will be downloaded as columns or appear as columns in the CSV file when you download your file. And note, for abundance view, you may want to pay special attention to this option here to include zero count samples. Some modelers especially find zeros to be very important. So check that box to include zero count samples, but of course this will make your download potentially significantly larger. And do note there's a link here, the data dictionary, which you can click on to go to a page that learn exactly what all the fields mean and information on what the terms mean that are inside those fields. And finally, when looking at this left-hand pane, you can click this share link button. I'm actually going to click here, and then I'm going to click the share link button. And this copies the link to your clipboard that can be pasted into another browser. So then you can send to your colleagues, for example, the view that you see on your screen when you're working with them and collaborating with them. So we find this to be a really helpful tool. Now I want to quickly switch over to a slide I made to show we really have a huge amount of abundance data in our map, which really kind of shows the power of what we're doing. This one. And here are some of the statistics on just how much we have for our abundance data. Over records on over 56 million animals collected, which comes to over 21,000 occurrence records. And you can see just how rapidly we've grown in the amount of data that we've curated and made available on our map. So I will go back to our map, clear all these filters, go back to seeing the world. And I'm going to switch now to insecticide resistance phenotype view. And I'm going to use the right-hand facet drawer again to add some filter terms. So I just want to see Anopheles gambiae. I'm going to switch to insecticides, facet by insecticides, and I just want to look for records that have deltamethrin used as the insecticide. And in fact, I only want to see data that's been collected using one particular kind of test, particularly WHO paper kits. So here now you see we have all the records we have on Anopheles gambiae mosquitoes that were exposed to deltamethrin using WHO paper kits. So we have phenotypic data on those results. So I'm going to click on this dark marker point here, which represents high resistance. And I'm going to look at our specialized graphs here. And as you can see, compared to the background from all similar data we have on our map, mosquitoes at this location have a lower mortality rate when exposed to deltamethrin. So they are resistant to the insecticide. And conversely, I can click on one of these blue points in Madagascar. And again, for the data that we have been given, I can see they have a very high mortality rate compared to background when exposed to deltamethrin, so they are very susceptible. And finally, just like everywhere else in our map, we can double click on a location, disaggregate that data, and on the fly, we can draw a different map, or draw a different graph. And once we get down to this level of data points, you can even highlight a given point. So this is a single assay that was performed and see all the metadata associated with that given point. And this view that you see right now with this pop-up is the same information that you would see in the table view. And you can see what kind of data that we have, what kind of metadata we have for insecticide resistance assays. So for example, we see that this was Anopheles gambiae sensulatu. It was tested on adults, WHO paper kit. It was exposed to 0.05% deltamethrin for 1 hour, and these were females, and we had 11% mortality. So it was quite a resistant mosquito. And again, also just like everywhere else in our map, you can download the raw data as a CSV file, or use the share link to share this view that I have on the screen with a colleague. I'll next switch to genotype view, and I'll clear my filters and zoom out again. And currently in VectorBase, the only genotypes that we have displayed in this map are those for insecticide resistance alleles. So here I will do a search for a filter for Aedes aegypti. Again, I'm not going to push enter, but click on the term that I want. I will go over to the right-hand drawer, and I'm going to do a facet by locus. And I want this KDR V1016, and I'll add that by clicking on it. And then I'm going to switch to my allele facet, optimize those colors to make the contrast work nicely. Click on a data point, and click on that pie chart, and I can see the allelic frequency of the samples we have at that KDR 1016 locus. And this is the voltage-gated sodium channel responsible for permethrin knockdown resistance. So at least for the samples that we have received, it seems that about half of the wild type and half are these resistant V to G mutation. I will switch to another view, one of our specialized views, pathogen view. Clear my filters. And here I'm going to search by depositor name. So I'm going to do the Southern Nevada Health District. And I see their data is, of course, in Southern Nevada. I'll click on it. And I just want to look at West Nile virus results. And I want to look at a timeline. And here we can see a bar chart, which is overlaid with a line chart. And the number of assays performed, so the pools of mosquitoes that were tested for the presence of West Nile virus, is represented by the bar chart on the left axis. And on the right axis is the number of the scale for reading the line chart, which is the number of positive test pools. And the reason why we have the two different scales is, as you can tell, oftentimes infection rate may be 1% or less. And so the only way you can do it is to have it on two different axes. And this is by year. So obviously this year was had a bigger West Nile problem, but we can also see higher resolution data or zoom it on a particular year with the slider. Excuse me. Finally, I will show you blood meal view. And this is our latest view, so it does have the least amount of data in it. And here I'm going to search by a particular author, Victor Brugman. And I'll search just for Culex pipiens data. Optimize the colors. And we don't have a specialized view for this. Oh, yes, we do have a specialized view for this data. I apologize. And here we can see the result of the blood meal host sources for Culex pipiens collected over time. So you can get an idea of what the different hosts of this mosquito are. And perhaps even estimate, and perhaps conclude that it might change in a seasonally dependent manner. And would be an area for further research that you might want to follow up. Now, one important feature, we have not quite moved from the old vector-based pop biomap to the new MapView yet, that I do want to mention, and will be restored soon, is that in the legacy site, which I show here, which you can still access by going directly to vectorbase.org instead of beta.vectorbase.org. If you go to the table view and you click on this little eye, you can see the provenance of any data point. And this includes the author and the publication, so you can know where we came up with this data. Now, I do know in both the legacy site and the beta site, when you do a data download, one of the columns that you will see is a citation information. So you can get that information is still accessible. So I have just shown you 5 different kinds of insect vector data views that we have in vector-based MapView. And as I mentioned earlier, we will be bringing MapView to the other VEuPath sites. Clearly, depending on which VEuPath site you use, the kinds of data that are collected, and thus our presentation of the data is going to look different. But I hope I was able to give you a taste of what the possibilities are. But I do think that things like the genotype view are universally accessible. Most of our users are working with genomic data. And so across the ViewPath sites, so it's very easy to imagine what kind of data we might show on a ViewPath MapView site for you. And we, of course, are working on more sophisticated ways of showing genetic variation, including multiple alleles across time and space. But many of you, for example, may work on resistance of your pathogens to various interventions, and thus my demonstration of our insecticide resistance view might be relevant in future versions of MapView. But please do reach out to us if you have any other questions or potential data sets or use cases that you believe could work well in both in vector-based or in the larger ViewPath MapView map. I see we have a question here. Let me see what it is.
You got the question, or are you going to read it out, or you got it? I think
so
you pretty much answered it, but yeah, go ahead.
Yeah, so there's a question on if one uses information from the database, for example, resistance information, how will one reference the specific work? Yes, and so as I showed, when you do the download feature, it will show you exactly which reference you should use to reference that particular piece of data. We, of course, would like you to also reference that you used the vector-based or MapView tool to locate the data, but do reference the original data providers and generators whenever possible. That's the only question we have. So I want to thank you very much for your time, and I'll pass it back to Omar for some closing remarks, unless Omar can think of any other things he'd like me to demonstrate.
I think the one thing I wanted to ask is you showed us the pathogen view. So what if a person has a specific pathogen in mind and I want to just explore, do we have any information about vectors with, let's say, Babesia or with Plasmodium? Is there a way to easily do that?
Yes. So, for example, filters, we can say Plasmodium falciparum, and we can go to the host species. And so we have records, they could be negative results, for all of these anopheline species that were tested for Plasmodium. So I could say I want to see Anopheles arabiensis, Plasmodium falciparum, and I want to only see the ones that it was found in there. Now, obviously, there are much more. We have much more data on U.S. viral surveillance, but you can construct queries like that, like I've shown. For show me this pathogen in this species that was found.
And so that's obviously a nice point. Again, as you mentioned, Sam, in the beginning, anybody on the call who's working on surveillance studies in Africa or other parts of the world, and you have access to studies that you think would benefit the community in general, definitely contact us so we can make sure we integrate the data into MapView. Great. So let me switch over. So thanks, Sam, very much for a great presentation. There's a thank you from some people as well. They enjoyed your webinar very much. So definitely, I'm showing here on my slide a link to our webinar survey. And we will go ahead and put this in the chat window as well. So you can scan this QR code or put it in. I'll go ahead and just put it right now quickly in the chat window so everybody has it. There, you got it twice. So please go ahead and answer this survey. In particular, let us know about what you would like for future webinars. We're also asking a question about running a virtual workshop, which we will be running in the future, but we're trying to get an idea about what kind of a virtual workshop you may be interested in. So please definitely go ahead and answer the survey questions. Also, another point I wanted to bring up is if you go to our homepage, in particular, in this case, vector-based, you will see, and I'm pretty sure Sam mentioned in the beginning, but we have some helpful resources, overview of resources and tools right in the middle of the homepage. One of them is a MapVEu card, which allows you then to read more, and it'll give you some hints and clues similar to what Sam showed you, and so you can get more information. In addition, you can access MapView straight from here, from this card, or you can go under the tools section and access MapView from the tool menu, and then it'll take you to the map that Sam was showing you, and you can then explore it further. There's an additional question that came up, which is what is the scenario of the study regarding mosquito species in India? And so, Sam, do you know if there's what kind of information we have for Indian studies?
Yes. Can you still see my screen? No.
I will make you the presenter one more time, and so that's easy to do. Okay, go ahead.
So we can also use the search tool. So I've switched back to sample view, which should show you absolutely everything. And you can search by large geography things, perhaps not cities, but definitely countries and states and counties. So we can always use more, but looks like we have some DNA barcoding studies, a blood meal study in that area. I know there's a isomer located in India, International Center for Excellence in Malaria Research, and they have submitted a large amount of the Indian data. So if I click, you can click on this button here, and we can go straight to abundance. And we can, for example, look at what was collected from this study. So yeah, we could definitely use some more, but we definitely have some in there now.
That's pretty cool. I like the reactive response, Sam, of the map. It's nice to click around and see how the graph changes as you move around from region to region. Great. Any do we have any other questions? Let's see.
I don't think we have any further questions.
Great. So this webinar was recorded, and so we will make this available on YouTube later today. You will get an email by tomorrow, which will provide you with a link to the YouTube of today's webinar. A reminder that we do have many additional webinars scheduled on a weekly basis, and so make sure you go to any of our websites. So if you go to VectorBase, for example, or beta.vectorbase or beta.plasmodb, you can go under the help section, learn how to use VEuPathDB and explore webinars. And this will show you a list. So for example, next week we have a focused webinar on VectorBase to demonstrate all the tools and functionalities of the beta site, beta.vectorbase.org. This will obviously be very, very useful for VectorBase users, but also useful for anybody who uses VEuPathDB resources, because we're built on the same infrastructure. So tools that are available in VectorBase can also be available in all of our other resources like PlasmoDB or GRDB or AmoebaDB, and so or TriTripDB or FungiDB. So those are all would be a useful webinar. And as you can see, future webinars include a webinar on searching phenotype datasets, orthology, and OrthoMCL if you're interested in using phyletic patterns to define species or genes of interest based on their phylogenetic profile. And then in July, we have one on RNA-seq searches in VEuPathDB, and we will continuously add webinars to this list for the foreseeable future. And so visit the site, this page frequently, and register for webinars that you find interesting. All right, it looks like no more questions. Thank you, Sam, one more time, and thank you everybody for joining, and see you online soon again. Bye-bye.